Why humans are not immortal. Hello, everyone, and welcome to today's episode. We are diving into a topic that blends science, biology, and touch of philosophy. Why humans are not immortal and how the Hayflick limit plays a key role in this. We will break it all and understand what exactly the Hayflick limit is. So, let's get started. The fascination with immortality. The idea of living forever is something that has intrigued humans for ages. From ancient legends of eternal youth to modern science fiction, the concept of immortality captures our imagination. But while we may dream of endless life, biology tells us that immortality is not feasible in the way we might hope. To understand, we need to explore how our cell works and age. So, first we have to understand basic cell biology. To grasp why immortality is not possible, we will understand a bit about cells. Our body are made up of trillions of cells, each with specific functions like forming our skins, muscles, organs. Cells are constantly dividing to help us grow, repair and maintain our bodies. There are two types of cell divisions. First is mitosis. This is the process where cell divides to create two identical daughter cells. Mitosis is crucial for growth and repair. And second one is meiosis. This is a type of division happens in a reproductive cell, sperm and eggs, and is essential for sexual reproduction. When cells divide, they copy their DNA so that each new cell has the same genetic instructions. However, this process is not perfect and has built-in limits. And the cellular clock, our chromosomes have protective caps at their ends called telomeres. Think of telomeres as a plastic tip of shoelaces. They keep the chromosomes from frying and losing important genetic information, right? And it plays a very key role because from our chromosomes have protective cap at their end that is called telomeres. And each time a cell divides, its telomeres shorten a bit. This shortening acts like a biological clock, marking how many times a cell can divide. When telomeres become too short, the cells can no longer divide. It enters a state known as senescence, S-E-N-E-S-C-E-N-C, senescence, meaning it is still alive but no longer function properly or create new cells. And the Hayflick limit is named after Leonard Hayflick, a scientist who made a groundbreaking discovery in the early 1960s. Hayflick found that normal human cells can only divide a limited number of times, about 40 to 60 times. And before they stop dividing, this number is what we call the Hayflick limit. And if these cells divided almost, let's say 60 times, it means approximately a human can live on a planet around 125 years. After that, human will die. This is what, in nutshell, is Hayflick limit. And you might wonder why cell cannot just dividing forever. Here are some key reasons for that. Because we are human, we always think a lot of things are pop up in our mind. So this is a very valid questions. Why cell cannot keep dividing? We always wondering this type of questions. So here are some key reasons we will discuss here. First is DNA damage. Now, how DNA damage cannot keep dividing cells? So every time a cell divides, there is a risk of errors in DNA replication. These errors can lead to mutations, which can cause the cell to malfunction or become cancerous. The Hayflick limit helps prevent by this stopping cells from dividing endlessly. If they are dividing endlessly, and if cells are dividing endlessly, it become converted into a cancer. So this is why the cells to malfunction or become cancerous. This is what the error. If error can lead to a mutations which can cause the cell to malfunction and become cancerous. So this is the one risk of that is called DNA damage. And Hayflick helps to prevent by stopping cells from dividing endlessly and accumulating too many errors. So the first reason is DNA damage. Second is energy cost. Dividing cells require a lot of energy. If cells could divide indefinitely, it would place a huge burden on the body's resources. This could lead to metabolic problems and other issues. Third one is aging and repair. As cells age, they accumulate damage and lose the ability to function and repair tissue. The Hayflick limit helps manage this process by ensuring that damaged or aging cells don't continue dividing or causing more harm. And another important concept related to cell aging is apoptosis. This is also known as programmed cell death. Apoptosis means programmed cell death. Apoptosis is a controlled process where cells are deliberately removed from the body. It's essential for various functions. Number one is development. During fetal development, apoptosis helps shape our body by removing cells that are no longer needed. For example, it removes the webbing between our fingers and toes. Second one is health. Apoptosis helps eliminate damage to potentially harmful cells. If apoptosis does not occur correctly, it can lead to disease like cancer, where cells refuse to die, or degenerative disease, where cells die too soon. And even if we could overcome the Hayflick limit, true immortality might not be practical or desirable for several reasons. There are a lot of reasons, several reasons. One of this, overpopulation. If people did not die, there would be an immense strain on our planet resources. We would face challenges like overcrowding, scarcity of food, water, and space. Second is aging without renewal. Without the Hayflick limit, our body would still suffer from accumulated damage over time. This could result in chronic health issues and a decline in quality of life, even if we did not die from natural causes. Third one is evolution. Death is an important part of evolution. It allows for the turnover of generations, introducing new genetic variations that help population adapt to changing environment. Without death, evolution would slow down, making us more vulnerable to new challenges, right? And while immortality remains out of reach, scientists are exploring ways to extend human lifespan and improve health. Here are some areas of research. Number one is telomeres activation. Telomerase is an enzyme that can add length to telomeres, potentially allowing cells to divide more times. However, this comes with risks, such as an increased likelihood of cancer, as telomerase is often active in a cancer cell. And stem cell research. Stem cells have the ability to divide indefinitely and could be used to replace damaged or aging cells. This research is promising but still faces many technical and ethical challenges. Third one is gene editing. Technology like CRISPR can potentially address the genetic causes of aging and cellular damage. And while this field is advancing, it involves complex science and ethical considerations. So, to wrap up, while the dream of immortality is appealing, biology sets clear limit on how long we can live. The Hayflick limits, telomere shortening, and programmed cell death all play critical roles in the aging process and our overall health. These mechanisms protect us from disease and help maintain balance in our bodies. Thank you for joining me today as we explore the Hayflick limit. And why we explore Hayflick limit? Because according to New York Times, Leonard Hayflick died at the age of 96. That is why we are discussing this concept here. And why immortality might not be the best outcome. We already discussed this thing. And I hope this episode have provided with you a deeper understanding of these biological concepts. If you enjoyed this discussion, please subscribe for more insights into the fascinating world of science and other tips. So see you next time. Thank you.